All right, good morning, everyone. My name is Derek Hall, and this is Cassandra Avila, and I'd like to recognize the other members of my team, Carlos and Javier, as well as our faculty mentor, Dr. Maria Alvarez. And today we are investigating the antibiotic susceptibility pattern and profile variation of the K12 strain of E. coli when exposed to both full gravity and microgravity. And we're going to be using Siemens automated Microscan to help us identify that. All right, so a little bit of background information. Now, previous studies done here on normal gravity do indicate that when cells are exposed to radiation, the most common form of damage is the deletion of certain DNA segments, and that deletion can result in a different gene expression. Now, however, the effects of radiation-induced mutations in microgravity are currently unpublished, which is why we opted to further explore this issue, and we wanted to use E. coli because it is a bacteria found in our intestine, and it is of importance to us. Okay, so once again, the objective of this study is to send samples of E. coli, the K12 strain, into microgravity and determine if susceptibility patterns of antibiotics were changed.
These are some of our methods. The E. coli K12 strand was grown in LB broth. We kept the suspensions at normal and microgravity conditions. And we analyzed these samples using the Siemens Microscan automated microbial identification system. This is more about the system. It's based on biochemical reactions. It provides antibiotic susceptibility data and displays the minimal inhibitory concentrations for commonly used antibiotics. We get the results in 24 to 48 hours. This was our instrument processing. First, we had to isolate the bacteria for the system. We had to do a gram staining test to determine if it was negative or positive. We did an oxidase test to then again determine if it was negative or positive. The machine gave us biochemical assays, and we compared these results to the database. It identifies the probability and species.
All right, so this is just a sample well that we use to inoculate the bacteria, which we insert into the machine, and it automatically identifies it. Now, once again, because it uses biochemical assays to help us determine what bacteria it is, some biochemical tests are displayed here, and once again, through a process of colorimetry using a spectrophotometer, different colors give us different readings. So for example, a glucose test would be positive by a strong yellow, a urease by magenta to pink, and hydrogen sulfate to a black precipitate. Okay, so for the ground control sample, once again, since we were working with E. coli, just to double check, we were able to verify that it was E. coli, a 99.9% probability of correct identification. Now, afterwards, the computer generates antibiotic susceptibility pattern, and it displays the commonly used antibiotics, as well as whether or not they're susceptible, and its minimal inhibitory concentration, which is measured in milligrams per liter. As we can see, since the K12 strain is a laboratory strain and is non-pathogenic, it is susceptible to some of the most commonly used antibiotics. Specifically, we wanted to highlight ampicillin as well as tetracycline.
So this was our experimental sample that was exposed to microgravity. Unfortunately, we were not able to recover the E. coli, but what we got back was Sphingomonas posimobilis, and we identified a 99.9% probability of correct identification. Then again, here is the same report again, but with the Sphingomonas posimobilis. Unfortunately, it was not susceptible to ampicillin, but it was to tetracycline and most of the antibiotics. These are our conclusions. The E. coli that we kept at regular Earth gravity conditions displayed normal biochemical reactions and antibiotic susceptibility patterns. We were unable to recover the E. coli from the sample exposed to microgravity. The bacteria that overgrew that E. coli was identified as Sphingomonas posimobilis, and unfortunately our project will be repeated. These are some of our acknowledgements. We want to acknowledge Jose Mendoza, Gloria Chando, Dr. Chavez, and especially Dr. Goldstein and the SSE program, and John Cosanto in the ITA program for letting us repeat our project.
So since you had the Sphingomonas take over from your E. coli, could you just rerun your ground control experiment with Sphingomonas? And so since you inadvertently have a microgravity sample of that bacterium, you could run a ground sample on that. Would that be valuable?
That could have been another way to approach the experiment. I don't think we actually thought about that, but due to the limitations of the laboratory, I don't think we had a strain of that in hand, ready to be prepared and suspended in broth for analysis.
When you do repeat the project, what will you be doing differently to suppress the bacterial growth?
Well, definitely we'd have more control over our samples to make sure that the E. coli we were able to recover the E. coli.
I'm not going to say this right, but this Sphingomonas that grew over the E. coli, do we know why it grew over the E. coli? Was it a gram negative versus a gram positive, or what allowed it to do that?
Sorry, we could only speculate as to how the Sphingonomus possimobilis overgrew the E. coli. You know, it's something may have happened during transportation, but once again, it's purely speculation.
What are the benefits of this experiment?
Well, the E. coli, we wanted to do E. coli because it was the bacterium that is found in our intestines. But what this is used for is you can see if the antibiotics if the bacteria is susceptible to the antibiotics. So say if someone gets an illness and all, we need to know what antibiotics to use to cure them.
What are you going to do next time to get better results or make sure that the Sphingomonas doesn't overgrow the E. coli?
Now with all the changes they're implementing, we're hoping that we can have more of a supervision as regards to how the specimen is handled. And in taking those precautions, we're hoping to keep the E. coli there and analyze that.
All right, thank you again.